Okay, so welcome back to part 2 of the cardiovascular physiology lecture, and we last left off talking about this diagram right here, and that's what we were going to get into next. So the reason I want you to think about it as a two-sided pump is because the heart does act completely differently with regards to the pressure systems and where the blood is going and coming from. Okay, even though everything is obviously connected as a one-circuit system, we have to look at the right and the left heart as two different entities at this point. Okay, so right here, just to follow the circulation patterns along, let's start with the left side of the heart. All right, so blood comes back in from the lungs. We just picked up a bunch of oxygen. We come in through this left atrium. We go through the valve here. We open it up, and then we increase the amount of pressure here to overcome the aortic semilunar valve. We pump it into the aorta right here, right? And then into the arteries and the systemic circulation. Okay, we go to the systemic side of circulation, which we go through the arteries, the arterioles, and the capillaries, right? So once we get down to the level of the capillaries here, here's where the venous and the arterial side of circulation meet, right here. Okay, so in these systemic capillary beds, this is where the O2 is leaving as we get dropped off at the tissue levels, right? So oxygen is leaving the arterial side, and then it is getting picked up by the venous side of circulation, and it continues on, right? So the blood continues on to the veins. You know, we first go into the venules and all that good stuff, and we go to the venous side of circulation to be returned to the heart. Why? Because it's been stripped of a lot of its oxygen, not completely, it's really never down to zero, but stripped with a lot of its oxygen, and we got to go back to the heart. So the right side of the heart, so that we can pump to the pulmonary circuit where the lungs are, where we're going to breathe in, bring some oxygen into the lungs. These capillary beds, again, here's where we're going to mix with the arterial and the venous side of circulation again. And then we're going to drop off oxygen into the arterial side of the circulation. Then we could bring pulmonary veins, bring blood back to the heart, right? Remember, veins bring blood back to the heart. Okay, and then we're going to repeat that all over again. Okay, so let's talk about elastic recoil and what that means. So when the left ventricle contracts, the aorta expands and receives this bolus of blood during systole. Okay, so the aorta stores some energy in its walls. During diastole, there's something called elastic recoil. That is when the energy, which is stored in those walls, compresses that blood and pushes it downstream, giving it some sort of pressure head that we can use during diastole, right? So blood doesn't just stop when we pump it out and then we rest and then pump and then rest. Blood doesn't just stop in the veins and the arteries and all that, right? It's still unidirectional flow. There's still flow during diastole, and this is one way that it does that. So again, during diastole, there's something called elastic recoil. That's when the energy is stored in these walls, right? So ventricular systole, we pump blood into the aorta. The aortic valve is right here. And then we shoot most of this blood downstream, right? But some of that energy is stored within these aortic walls right here. And that's advantageous to us because during diastole, that energy is going to act as a second pressure head to keep unidirectional flow and flow moving in one direction. Okay, so therefore, when we relax, we still have constant flow. Blood flow does not stop during each diastole, like I just said. It's constantly moving, right? So even when we're at rest or our heart's at rest, blood is still moving throughout the system. It doesn't all just stop. So that's our first factor, helping blood going from pulsatile flow to steady flow. Remember, we want steady flow down at the capillaries in order for efficient gas exchange to occur. Okay, so let's talk about vessel anatomy and function. So blood moves rapidly through the aorta, right? Because it was just ejected from the left side of the heart and its branches. So it moves very fast through the aorta and its branches. The vessels or tubes get narrower and thinner the further we get away from the heart and the aorta to the periphery, right? Again, so we go from the aorta, we branch into larger arteries, and we branch into arterioles, and then we branch even further into a capillary network. And then once we drop oxygen off at the capillary level, and we pick up some waste, right? This connects us to the venous side of circulation, and we go from small branches to venules, to veins, then to the vena cavas, right? So we go from large we get a little bit smaller, but we're still large arteries. Then we go to arterioles, which are a little bit smaller, and then we go to the capillaries, which are very small and multitude. So as we branch down, we multiply the amount of vessels that are given with each different section, right? So arteries, there's fewer arteries than there are arterioles. There's fewer arterioles than there are capillaries, right? So there's a multitude of capillaries branching off of each arteriole, and there and so forth. Then we go the same way opposite. All right, we have multiple venules, then we go into the larger veins, and then we go into the large vena cavas. Go back to the heart. Okay, so the vessels and tubes, like I said, get narrower and thinner the further we go away from the heart. The aorta is mainly made up of elastic tissue. So the aorta is mainly made up of elastic tissue. This has a good amount of elastin stored in its walls. This is important because we are ejecting a large bolus of blood from the heart, and this vessel needs to be able to expand to receive this large amount of blood. As we move to the periphery, the amount of smooth muscle in the vessel walls increases. Okay, so we don't have a lot of smooth muscle in the aorta or arteries. We need them in the arterioles. Okay, so we need the aorta and the arteries to be much more elastic in nature because we have just ejected a large amount of blood in. It has to be able to accept that blood in. It has to be able to expand as well as compress that blood downstream. Okay, so let's talk about the arterioles. Okay, the dominant tissue of our arterioles is smooth muscle. These vessels offer the greatest resistance to flow. There is a large drop in pressure that moves from the arteries into the arterioles. So there's a large amount of pressure going into the arterioles, but there's a low pressure coming out of the arterioles into the capillaries, right? So ultimately, pressure is an energy form. We need to have a larger pressure going into the arterioles since those are the vessels that offer the greatest resistance to flow. The blood needs to push through a higher resistance. So therefore, we're going to have low amount of pressure coming out of the arterioles. In the arterioles, we can just adjust the degree of contraction of that smooth muscle, right, in those type of vessels. So by adjusting the degree of tension in the smooth muscle, we can shunt blood from one anatomical location to another anatomical location. So we're moving blood from areas of lower activity to areas of higher activity. So this means now we can be an exercising human. By changing the tension of the vascular smooth muscle, we change the amount of resistance that we have to overcome to maintain flow. This resistance of the arterioles is another factor that allows us to convert flow from pulsatile flow into steady flow. Okay, so let's look back here at the picture of the arteriole right here. This is the vascular smooth muscle that surrounds the arterioles. Now this vessel, the arterioles, offer the greatest resistance to flow. So flow coming in is going to be higher from the arteries, but then because this vascular smooth muscle can constrict and relax, the more we're constricted, the more resistance we have to flow. So what's going to happen to flow when we reach the end of the arterioles or into its branches, right? It's going to be much slower. And that's advantageous to us because what's happening here again, we have gas exchange. We need it to be slow. So this is a positive thing for us in order for gas exchange to occur, because if blood still was just super fast going through these arterioles and its branches, then we wouldn't have enough time to exchange oxygen and pick up waste. Okay, so let's do a quick summary points of what I just said. So from going from pulsatile flow to steady laminar or smooth flow in the capillaries, right? There's two factors. Our first factor is that elastic recoil of the aorta, right? So we store that energy in its walls and it acts as a second pressure head during diastole. So that's number one, because we keep flow going during diastole. So it doesn't just stop and go. Second factor is when the blood hits those arterioles, everything slows down because of that vascular smooth muscle. So it slows down enough so that the next pressure wave can catch up to the preceding pressure wave. Okay, so once we slow that blood flow down in the arterioles, that preceding pressure wave can catch up to that pressure wave that is there, right? So then that's going to be our second factor that makes kind of this pulsatile, noisy flow into this steady laminar flow, which we need at the level of the capillaries for gas exchange. Right, so we can see here, like we talked about, the left side of the heart and arterial side of the circulation, and then the venous side of circulation, which is completely different. So when we look at the pressures throughout circulation here, we can see that they're vastly different, and they need to be. So this is why the right side of the heart and the left side of the heart are completely different. But these pressures need to be different. Why? Because we need that unidirectional flow, right? So remember we talked about in that very first lecture when we talked about gradients and how pressure gradients is one of our governing gradients in our body. This is where this is really coming into play here in that concept. So remember, flow goes from an area of higher pressure to lower pressure, okay? So right here, this is where we generate the highest amount of pressure, okay? We generate about 120 millimeters of mercury, right? We pump out into the left side of the heart, we pump out into the aorta, we go into the systemic arteries where the average pressure in the arteries is 90, right? So we're going from higher pressure to now lower pressure because we're going into smaller vessels. So we're at an average of 90. Then we continue on, right? And then we go on to the arterioles, which is a little bit less pressure. And then now we're in the capillaries where we're at about 20 millimeters of mercury pressure. Then we continue on, and now we go to the venules and to the veins. Now we're at 10 millimeters, right? So it has to, in order for us to keep moving, we got to have high areas of higher pressure to lower pressure. So we keep moving up. We move up to the right side of the heart. This is the right atrium. So the right atrium has 3 millimeters of pressure within there. We need to have that pressure difference of that 10 millimeters of mercury from the systemic veins into the right atrium for blood to move. So then we go down into the right ventricle, and then what happens? We close that valve, and then by closing that valve, we're going to generate a good amount of pressure within this ventricle. We're going to only generate about 25 millimeters of mercury as compared to 120 on this side. Why? Because like I said before, the right side of the heart is a low pressure system. So when we pump, we generate 25 millimeters of mercury, we pump out of the pulmonic valve into the pulmonary arteries, taking blood away from the heart to go pick up oxygen, right, in its lungs. So in the pulmonary capillaries, we go from the pulmonary arteries average of 14 down to 12 down to 10.5, and then we come back to the pulmonary veins. We have 9, and we just have a smaller gradient to come in, and then boom, we do it all over again. So you see how these pressure gradients are key in order for us to keep blood flow from the left side of the heart all the way through the body to back to the right side of the heart. But these pressure differences are key in understanding flow of the heart and the circulation. Okay, so to sum up here from these different branches, we're going to talk about this capillary network. So many capillaries branch off of the arterioles. So we have a very wide capillary network relative to the dimensions of each individual vessel. So the network itself is very wide, but each vessel is very, very small, very, very narrow. So the vessels themselves are very small in diameter individually, but the amount of vessels gives us a large, vast network. So we have a very large cross-sectional area when we add all of those up together. But each individual tube is very small. So we increase our vascularity as we move more to the periphery, right? Because as we move from the aorta to the arteries to the arterioles to the capillaries, we progressively get more and more vessels that branch off of each one subsequently. Okay, so blood flows down even more into the capillaries. The capillary walls are only 1 cell thick. So that's, if you can get a good picture there, the capillaries are very, very small. Their walls are only about 1 cell thick. So this is important with regards to gas exchange. Okay, this gives us time for gas exchange to take place. So as we leave the capillary beds, we then head into the venous side of circulation. But it's very important that only 1 red blood cell can go through a capillary at a time. Why? Because then that means it's nice and tight going through the capillary. And if the capillary wall is only 1 cell thick, then we are more efficient at exchanging oxygen and carbon dioxide, so nutrients and waste with one another, to go then to the venous side of circulation. Because if it was very, very wide, then we wouldn't be as efficient as dropping off oxygen onto the tissue level. So it's very beneficial to us that the capillaries are 1 cell thick. Okay. Okay, so next we are going to talk about 4 major changes that take place moving from the arterial side of circulation or systemic side of circulation to the venous side of circulation. We're going to pick that up on the next video.